So welcome to the second lab. My name's Philip, if you haven't met me already. And so today, the outline of things that we're going to cover are STAR, and with that comes SLURM, also RCQC, and then RSAM and SAMMON. So these are all things that are going to be very important for completing the homework. Like I said, feel free to go onto Canvas under Module 1 and download the slides. And also, please SSH into Cannon because at some point I'm going to ask you to try something out just so I can teach you guys like how to start a job on Cannon. Yeah, and I think the focus is going to be on like running these as opposed to like what the algorithms do, because that was kind of covered in lecture. All right, so let's just start with STAR. It stands for Splice Transcripts Alignment to a Reference. And so if you're in lecture today, you'll see that when you're doing RNA-seq, the reads that you get, they aren't coming from contiguous locations in the genome, right? Because when transcription happens, there's exons and introns on a gene. And then these introns get spliced out, and maybe these exons get permuted in some way, or some of them get thrown out to produce the mature transcript. And so when you're doing alignment on RNA-seq, you really need an aligner that realizes that the reads are going to be that way. And so like Professor Liu said, STAR is probably the state of the art for splice-aware alignment. And so I won't get too much into the details of the algorithm, but I think this figure really gives a nice like overview of what it's basically doing. So like if your reads up here, and then the gray parts can be thought of as the exon for which the read might have came from, or the exons of a gene. So STAR, for example, STAR is okay with just mapping the first half of the read to some part. And then if all of a sudden the rest of the read won't map, STAR will say, okay, I'll look later on to see if the second part of the read can be mapped. So like the first half maps to this gray region and the second half maps to this gray region that's possibly separated by an intron, which could be fairly large. Yeah, so I mean, STAR, I guess it uses a suffix array to kind of, the way that you can think about this is what STAR is basically doing, it's building like a humongous hash table so that it can efficiently take in an input read and then find where in the human genome or where in any genome that this read came from. I mean, so what you have to know, especially for this homework, is that STAR aligns very fast, but the cost of it being fast is that it's going to require a huge amount of memory. For most mammalian genomes, STAR is going to take probably 30 to 50, well, maybe 20 to 40 gigabytes of RAM to kind of read in this huge hash table. And so this is why we're making everybody do the computation on Cannon, because we don't think that most people have that kind of resource, like on a personal laptop, for example. Okay, and so like if you have the raw slides or the file like on your computer, it might be nice to browse through the reference manual for STAR. It will show you how to do a lot more advanced things that kind of won't be covered here. Okay, yeah, so I'm just going to briefly talk about Cannon. I mean, I know Martin probably covered that last week, but I mean, basically Cannon is a supercomputer or a cluster, which basically means that it's a lot of small computers called nodes, which we call nodes, and they're combined to work together as kind of one giant computer. So what you need to know is, in each of these small computers are going to be doing a different task to kind of serve the good of the larger computer. So there's kind of basically two kinds of nodes that you'll encounter on Cannon. The first is login nodes, and these are the ones that you go to when you first SSH into Cannon. And so you can tell if you're on a login node by looking at like the thing after the @ on your username. So this says Boston, this stands for Boston login 3. So it's probably the third node in Boston, and it's a login node. And what you have to know about these is these basically serve just to get you on the cluster and allow you to look around at different files. But you really shouldn't be doing any intensive computations on the login nodes. And in fact, it probably will stop you from doing so. It's also about being a good citizen of the cluster, since if you're doing heavy computations on the login node, it's going to make the login node slow because it's not designed to do that. And it's going to make Cannon slow for the other users. So really, if you're going to do something like star, you need to go to a compute node. And so the point of this Slurm that I'm going to talk about is it's going to get you from the login node to the compute node so that you can run things like star. So how do you go from a login node to a compute node? Well, first, you need to specify what you want. So for example, you might say, I want 500 gigabytes of RAM and I want 1,000 CPUs, right? That's obviously quite a lot of computational power, and Cannon has enough to do that. But if everybody asked for that all at once, then everything would get filled up and there'd be, I guess, no room for anybody else to do anything. So basically, Cannon has a job manager called Slurm that, I guess, manages the user requests. And for example, like it will decide when to give you what job, and how it kind of like ranks all the jobs, deciding which one gets to go when. Yeah, so for example, and this might be something that you take down. So like I said, for star, you really need 10 to 20 gigabytes at least of RAM. So to be safe, if I was doing this problem, I might say, I want 64 gigabytes of RAM and I want 16 CPUs, and I want it for 2 hours. You also have to specify how long you think your job's going to run. And one way to do that, so there are kind of two ways to run jobs. Today, I'm just going to talk about interactive jobs because I think they're a little simpler to start out with. So the command here is srun, and that stands for slurm run. And then so the rest of this is just specifying what I want. Nodes and number of tasks per node can always be one, probably for this pset, because I'm only going to do one job really. I'm just going to run star on one sample. Mem, that stands for memory. CPUs per task, 8. That seems to be a good number. 16 is also possible. Time, this 2 hours. And then I want a bash shell. And then the -I stands for interactive. So that means it's going to give me a terminal on the compute node for which I can type in commands like interactively. So for those of you, okay. So if I type this in and I press enter, then first I'll see srun. This job, it has an ID number, has been queued and it's waiting for resources. Next, it gets allocated the resources. And then all of a sudden I go to I have this new, I guess, ID for what node I'm on. It says Holy, and that stands for Holyoke. And it's not a login node. So I've successfully gone to the compute node. And so, right, this can take probably a moment or two. So what I want you guys to try is, so for those of you who are on Cannon right now, I was hoping you guys could put in this job, which is a very tiny job. It's only asking for 8 gigabytes of RAM for 10 minutes. So can you guys give that a shot? Also, I can't see the chat right now. So maybe Jack, if there are any like questions in the chat, you can like relay them to me.
Sure.
Is this working? Has anybody tried this yet? Is this working? Would you like me to do it too?
Yeah, that would be helpful.
Okay, great. So let me just, oops. Are you still seeing my screen?
Yep.
Yes.
So are you seeing a terminal now?
Yeah.
I need to copy. Okay, let's try this. Okay. So I'm on, so I'm at a login node. So let's copy and paste this in. And let's see what happens. Okay, I press enter. Okay, so it's queued and now it's waiting. So okay, great. Awesome. So it worked. I don't know if it was going to be slow because everybody's asking for it. So you see now I'm not on a login node. I'm on a node called C's Micro 3. And so now like, the same, I can still do the same commands, ls, all the files are still there. I can still go to the stat115 folder. And it's slow for some reason. But yeah, so I think, yeah, so this is how you start a job. And yeah, so it can be kind of slow sometimes. There it goes. But yeah, and so since I only asked for 10 minutes, what will happen after 10 minutes is it'll just shut down, even if I was in the middle of something. And so make sure you specify enough time. And so to get back to the login node, I just type exit. And then the job's done. I can also type saccount, S-A-C-C-T. And it shows a history of all the jobs that I've asked for. So the one that I just asked for is completed. Yeah, any questions about that?
I had a quick question. If you were done before the time you allocated and you type exit, does it kind of free up the resources, or do you have to be really like mindful of exactly how much time you ask for?
No, I think it frees up when you exit. What you have to be mindful of is if you ask for like 20 hours, it's going to sit there queued for like forever.
Okay.
So that's the main factor when you're considering how much to ask for. You want to ask for enough, but really not much over. Otherwise, you're going to sit there forever and wait. Any other questions? Okay. So let's show how we can actually run star then. Okay. So I'm going to give you like an overview of the steps that I might do to run star. So step one, obviously, is to start an interactive job, or you can do a batch job, but we can talk about that a little bit later for those who are interested in batch jobs. So first, I start the interactive job. I go to my compute node. I've asked for enough resources, I've asked for enough time. Step two, this is all step one. I also make sure that once I'm on the compute node, I go to my home directory. I'll talk a little bit more about this later. But basically, we don't want you writing any files to the stat115 folder. So just make sure you're putting it in your personal directory, and you can go to that by typing cd home with this $sign. Next, you're going to load the module. You're going to load the star module. You can think about this as like in Python, you might import pandas, for example. In R, you might load a library by calling library. This is the same idea. You're just going to load, I guess, star onto the compute node. And the basic command is literally just to type star. But then obviously followed by star, there's a lot of different options that you need to specify to make sure that it does what you want. Okay, very quickly, like I said at the beginning, star accomplishes its speed by creating this kind of huge hash table for the human genome. And so I want to briefly tell you guys how we made this. This is actually not required since we've already done this step for you. And it also takes like several hours. But basically, after you type star, you can type -- run mode genome generate. This is going to like generate, I guess, the suffix array for the genome. And then you have to specify, okay, a genome FASTA file. So that's like a FASTA file is like the raw sequence of the human genome. And then you also have to give it like annotations to say like where in the sequence are the known genes and so on. So if you want to try this, it's totally optional. But I've put the files in HG38 raw, the raw like human genome sequence, if you wanted to try making the index yourself, but it's not required. So let's talk about the thing that is required, which is to perform the alignment. And so the basic command is going to look something like this. You type into your, I guess, you type into Canon star, and then -- genome directory, which is index path. Or genome directory, this is the path to the genome index that we made in step 2. And we saved it here at stat 115, homework 1, star HG38 index. So that's what you'd put here. Next, the read files in. These are where you put your FASTQ files. And so if it's paired in and you have 2 FASTQ files, you can just separate them by a space. So you can put 2 FASTQ files here. And then finally, you can think of threads as like, how many things at once are you going to be able to do? So like, if you can do some things in parallel, then it's going to go faster. And 4 or 8 seems to be a good number for this assignment. So I got this to run in around 15 minutes with the resources that I've asked for. I guess any questions about, I guess, this call?
So we would run the first like basic star command and then this.
This should, so like, oh, no. So what I meant by this is that the basic command to do anything in star is star, but then you have to, it has to be followed by options. So what I meant is like, if you want to do run mode genome generate, then you type star space -- genome generate. If instead you just want to do alignment, you type star space. There's no, the default mode is alignment, so you don't have to say anything, but then you just have to specify where the index is and where your FASTQ files are. Yeah.
Okay, that makes sense. Thank you.
Sorry, what was the run thread and 8 means again?
So threads basically can be thought of as, so if you have 8 threads, then you can do 8 things. The computer can do 8 things at once. And so that's kind of helpful in this situation because like, I don't know exactly how star does it, but presumably you can parallelize some of the work and then you can do like 8 things at once. I guess like you can just like split all the reads into 8 groups and do them in parallel. And so that will make it run faster. Yeah, so that threads and CPUs are kind of the same thing, but not really. But you can also think of them as like cores. Yeah.
Okay, thank you.
Sorry, do you just type in that command to load star? When I paste it in, it says command not found.
Module load this didn't work.
Oh, I didn't include the module, I think. Yeah.
Yeah, sorry, yeah. So module load, yeah. And if you don't know what star is called, you can always type module query and then like some string. Like if you didn't know like what version you wanted, you can always type module - query star and it'll tell you what versions you have available. Okay, so Step 4 is to view the results. And so I guess star creates output files that will be useful for answering problem 1 in the homework, which I think just asks you to like say what % of reads are mappable, which % of reads are unmappable. So I guess I have a question for you guys. Suppose that I have a file. It's like, suppose it's a text file. I guess, how can I view it, or what's the easiest way to view the contents of that file on Canon? Can anybody give me the command?
Less.
Wait, like LESS?
Yeah.
Oh, does that work? Okay. Yeah, I mean, I was thinking about cat. Is that the same thing?
Yeah, actually, I think last works, but let me confirm.
Yeah, I mean, honestly, I don't use.
Less gives you a scrollable buffer. Cat prints
Oh.
everything.
Okay, so maybe cat's slightly easier for small files. But yeah, okay. Okay. But you can also try that, or you can even open like a text, an editor. Yeah, but that might be useful. So for example, I think we ask you to copy this report and just paste it into your R Markdown file. And so you can just print the contents, copy and paste it over to your R Markdown. Let's see. Yeah, so for example, like include like your bash command, which is basically going to be like one line where it tells you to. All right. Common mistakes that I just want to talk about briefly. So if you don't specify the time, then like I said, it's going to shut off in the middle and it's not going, and you're not going to get the results. So specify at least 30 minutes. I like to specify 2 hours just to be safe. I think if you choose run thread to be larger than CPUs per task, you might get an error. I'm not actually positive about this. Generally, you want to choose threads to be a little bit less than the number of CPUs you requested. And the final error that hasn't really been an error is, so we actually don't want you guys to save the results to stat115 folder. We want them to save them to your home folder, and that's for a variety of reasons. One being that it basically gives other students the answers if you save it to the stat115 folder. And so I guess any files we see in there will be deleted. So yeah. Okay, so any last questions on star? We can do some.
I had a question, sorry. So just to make sure, but we'll be running this through RStudio, right? Not just in the terminal.
I guess the HTML file that you're going to submit is generated with RStudio. You don't actually have to run. So you can put like eval equals false in your RStudio R Markdown file. So you're not actually going to run the bash command locally. You just show us what you did.
I see. Okay.
And if it looks reasonable, yeah.
So we only need to submit the results of running star, not the commands we use necessarily to run it, or both?
No. So I'm saying you have to submit. So you have to like copy and paste, say these are the commands that I use to run it.
Okay.
But we're not going to there's no way to run it locally probably. And we're not going to run the bash either. Does that make sense? It's like we want the commands, but you can put like eval equals false on the chunk. So it doesn't actually run when you try to knit it.
Okay, perfect. Thank you.
Yeah.
I have another question.
Sure.
How do we save the new file to our home directory? Because like when I run the star command, it says that I'm not able. I don't have permission to write in the directory, so I don't know where to.
So are you? That might be because you're trying to write to the stat115 directory.
Yeah, I know, but how can I change that?
So the easiest way is to run the command star in your home directory. And then when it's time
Ah,
to specify
okay,
the
okay.
genome directory, you give the absolute path, which is here.
Okay,
Yeah,
okay,
so
okay.
that's what I
Thank you.
recommend. Yeah.
The output of star. So one of the questions on the homework, it asks how many reads are mappable.
Mhm.
Does that include uniquely mappable reads, reads mapped to multiple loci, and reads mapped to too many loci? Like all three of those?
Yeah, so I think, so are there any reads mapped to too many loci?
I got that there were some. Like
Oh,
it's a relatively small number, but that there
Yeah.
were some. And I wasn't sure if like if it's mapped to too many loci, whether that counts as mappable.
yeah, let's say that's mappable. Also just say like, I considered mappable to be reads that are mapped to too many loci, any reads that map to any number of loci.
Okay, perfect.
Yep.
Thank you.
So let's talk about RCQC. So mRNA is kind of an unstable molecule. It's kind of just made to like leave the nucleus and go to the ribosome, I guess, to make a protein. And so it's not actually made to live for a long time. And so one important thing is to check to see if your sample, if the molecules in your sample have been degraded before performing further analysis. So I think Professor Liu's example is like, if you just have like tissue stored in a freezer, you probably want to check to make sure that like the sample hasn't been degraded to the point where you can't do any further analysis. And so RCQC is I guess it's a Python package. It's also like a collection of Python scripts that give you some measure of your sample and transcript quality. And so for problem 2, the two files that you want to focus on are this tin.py, which is going to compute a transcript integrity number, which is a measure of quality for each transcript. Then to like assess the quality of a single sample, you just take the median across all transcripts in each sample, and that gives you like a measure of the sample quality. Tin.py does this for you automatically. The second one is genebodycoverage.py, which makes the gene body coverage plot. So let's take a look at an example of a gene body coverage plot. This will actually be output by genebodycoverage.py. But so I guess my question to you guys is, so which samples have been degraded? I'm gonna guess why. So on the x-axis, I guess it's like different, like a list of the base pairs in a gene. And on the top, on the y-axis, is how many times each base pair was covered.
So probably all of the ones that are leaning more towards the right have been degraded.
Yeah, so like, it's probably not a good sign if like, always when you look across all genes, if like towards the 5' end, you don't see many, I guess, nucleotides in that transcript. That's probably not a good sign. That might indicate that you have like really some sort of serious degradation going on. What you want is like kind of like a flat, kind of have even coverage across all the genes, at least in the middle section of the gene. And so these are kind of what you're looking for on the gene body coverage plot. Yeah, and we'll talk a little bit more about that in a second. But so, installing RCQC is a little bit tricky. I don't know if you guys saw my Slack post. I responded to somebody. And I suggested this as a way to install RCQC. So basically you load Anaconda, which is like, I guess, Python and Conda, which is a package manager. And then you create a Conda environment like this. To activate that environment, you type source activate environment name. And then when you're in that environment, you can type conda install bioconda rcqc. And this should be writing files to your home directory. And so then the relevant Python files are contained in, this is the path to find like tin.py and gene body coverage.py. So this is a little bit complicated. Has anybody tried this yet and had it work?
Yes.
So I'd recommend maybe if you have Canon free to start initiating this process. It takes like 20 minutes. Yeah, so Conda is basically like a Python package manager on Canon. It can also load at non-Python packages.
Excuse me, if we do this for the current session, will it always be installed in our Canon like home directory, or like will we have to do it every time we want to use a Python?
Yeah, so once you do this, it's going to be there forever in this folder. But when you actually want to run it, you're going to have to do module load Anaconda and then source activate the environment. So like, I'll explain this in a little bit, but like when you go to your compute node, you're going to want to activate the environment before you run the file. And so I probably have like an instruction.
When we
Yes.
module load again, do we have to specify the 5.01, etc.?
Maybe. So if you just type module load Anaconda, so there's like a default version that will load. No promises that this is the one that, like, for example, I tried it with. But it will work. Something will load if you just type module load Anaconda, followed by source activate the name of your environment. This is your choice.
Thank you.
Yeah, and so then you can actually call, like, you can type Python space path to the tin.py file. Just the standard way to run a Python file.
And does it have to be Python 2, or can it work with Python 3?
So if you type Python 2.7 here, then when you call tin.py, you want to type Python. So you'll have to check, but I installed it 2 ways, and we can talk about this maybe at the end. If this way you don't like, you can also install it some other way in which I got it to work with Python 3. But this way I would just say Python. Yeah, Python 2.
Okay, thank you.
Yeah, it's a little, this is probably the hardest thing to install on this piece. Sorry about that, but hopefully this makes it clear. Okay, so let's just talk about how to run tin.py. Once you get it installed and you've activated your environment, basically it's, Python space path to tin.py, followed by all the options that you need to specify for tin.py. And so I've written down the relevant ones here. So a -i, these are your input files. And you'll see that, so we want you to run it on like 4 different samples that are all contained in a folder. It's under raw data 2. I think it's called BAM files, might be the name of the folder. And we want you to run it on every BAM file in this folder. And so you can see if you read the documentation here that you can just specify a path to a folder that contains 1 or more BAM files. So that's what you can do for this. I don't know what minimum coverage means exactly here. It's not super important. You can use the default value. But what is important is that you specify a reference gene model. And what I mean here is that, like, when you go to make your plot for gene, oh, this is for tin. It's the same idea though. So like tin.py will compute like a measure for each transcript. But so what you're going to tell it here, sorry, is to only use a small subset of genes. So you're not going to check degradation on like every gene that you could possibly think of. You're only going to test degradation on a select number of genes. And these are housekeeping genes. So does anybody know, like, can anybody give me like what a housekeeping gene is? Anybody heard of that?
A gene that is supposedly the expression will not vary a lot.
Yeah, so like a gene that's kind of reliably expressed under normal conditions can be called a housekeeping gene. And so it really speeds it up if you just specify that you want to, and this is good for assessing quality because, right, if you have degradation everywhere, then you probably have degradation on the housekeeping genes. So it should be sufficient just to check these. So we actually stored these housekeeping genes. They're a .bed file in the raw data 2 folder. So if you guys go there, you'll see it's called, like, it has housekeeping genes in the name. So it should be easy to find. Any questions about this?
So I guess when we run this package, we just specify ref gene equals the file path of the housekeeping gene in the data folder, right?
Yep. It's a .bed file, yeah. Yeah, it has like housekeeping gene in the name.
So when we're running these commands, can we either type -i and then the path to the input file, or -- input equals the path to the input file?
Yep. So you can type -i space input file, or -- input equals.
I see. Okay.
One's just shorter.
Then the actual like full command for running this would be tin.py space Python space all of these different flags? No.
No, you have to put Python first. Python space path to tin.py, then all the options.
Oh, okay. And it just knows to use tin.py because you gave it the path.
Well, like the path ends with, so like after bin, there'd be a /tin.py.
I see.
Yeah.
Okay, cool. Thank you.
Yeah. Okay, so I mean, quickly, gene body coverage, and it might have an underscore. It's the same idea. You can use default values for whatever this mRNA length is. Still, you have the same reference gene model. But you do want to call modulo R before calling the script, because kind of strangely, it's a Python file, but at the end, it writes an R script and tries to run the R script because it's going to make a picture like this. So just make sure that you call modulo R before doing gene body coverage. But it's the exact same idea as tin.py. Yeah, and if the question is, what should I do for problem 2? I think 2 plots or 2 charts would be sufficient. One is like, here's the tin score, median tin score, and maybe some standard deviation for each of the 4 samples. And then also, if you can make this plot using R, the gene body coverage plot, that'd be enough. Followed by like a few sentences of like what you've done. Because I think you're pretending that you're explaining this to like one of your collaborators. Okay.
Could you explain how to make the plots again using R?
It's going to do it for you automatically. So as long as you have modulo R before you run genebody coverage.py, at the end, you're going to get something. It's going to be a file that's a .pdf. And here you can change it to PNG or JPEG if you want. But it's actually going to make it automatically for you. So as long as you can run genebody coverage.py and you have R loaded. If you don't have R loaded, it will say, I don't know what R is, and then it will error. But you'll still get the raw data. Does that make sense? So it's just going to do it automatically. It's going to automatically make the plot.
Okay, and the plot for the 10 scores, we have to make that ourselves, right?
Yeah, so I mean, there's nothing in particular we want. I mean, a table, maybe a bar plot.
Okay.
Yeah, I think, and maybe say like, you know, a 10 score can be used to measure sample quality. And then we did that for each sample. I guess like include a few English sentences instead of just like code and figures.
Okay, thank you.
Sorry, could you go over one more time how to run the tin.py? Like, where does the path directory to the file go, like before or after writing out tin.py, and then the specifications for like input file, reference gene model, etc.
Yeah, so let me, so Python goes first. So it's just like, you know, anytime you're running software, you generally put the software name first. So like star would go first. Here, Python goes first. When you call Python, type Python, you always want it to be followed by a Python file. And so second, after you type Python, you want to put the path, you want to put tin.py, but it's possible that tin.py lives in this folder that's like 5 subdirectories down from home. So you have to just make sure you specify the location of tin.py, because if you just type Python tin.py and you're in your home directory, it's going to say, I don't know where tin.py is, and it will throw an error. After you do those two things, then you put your input file, so -i input files. And if you read the documentation here, it says you can give the path to a folder containing one or more BAM files. And so we specified the location of that folder on the homework. And so that's your input file. Next, you need to specify the reference gene model. This is what genes you're going to use to actually compute the tin score. And you don't have to do anything with -c. So I think just -i and -r are needed. Yeah.
Okay,
Does
got
that,
it. Thank you so much.
if we have some time at the end, I'll actually get it out and do it. Or at least like show you where tin.py lives, because it's a little bit confusing. Great. So what's here? Okay. Okay, so something that I'm definitely probably going to do next time, or maybe at the end if we have time, as a demo. So I like to only use Canon for computation, and I really don't like working on it. And so what I like to do is every time I run something, I get the output files. I just transfer that to my local computer, to my like desktop, for example. And then I run like all my programs that are installed on my local computer. And so you want to know how to transfer files from Canon to your computer. And potentially the other direction too. In case you like write a script like on your local computer and you want to send that to Canon so you can use it on a bigger data set. So the way that you copy, and this probably only works for Mac OS and Linux users. But basically I can open a terminal on my personal computer, and then I can type SCP, this stands for secure copy, just CP is copy. And then, so I type, I guess, this is like my login.rc.fast at Harvard EDU, : the path to the file on Canon, space, followed by the destination path, where I want it to go on my local computer. So have people figured out how to files yet? It's kind of a tricky thing.
Actually, Philip, is like if you don't want to do SCP every time, like you can sort of install an app called FileZilla, which will like have a sort of an interface.
Oh, really?
Well, I'm not sure if I can share the screen.
So can you show this at the end, like once we have it?
Yeah,
kind of like
yeah,
more
I
open?
can. Yeah.
And then I could also do a demonstration of this. You can do a demonstration maybe,
Sure.
or maybe Thursday, because we'll have a lot of time on Thursday. So
Mhm.
I still want to cover Salmon and RSAM. But I think this is the basic command. I can send a link in the chat, which like gives you a guide on like how to use or how to transfer files both directions. But it's you'll probably want to do this for problems 2 and 3, or just problems 2, or just problem 3, and problem 6. You might want to transfer files to your local computer. So it's good to make sure you know how to do this. Quickly, I'll give some tips for problem 3. So the text file that gene body coverage will output, I've noticed that they're not standardized to be between 0 and 1. And so you'll see that these gene body coverage plots, they normally go from 0 to 1. And you do that by, I guess, for each sample, you divide by the maximum number of reads or the maximum coverage at any like position on the, or any percentile on the gene body. So just make sure you standardize your values to be between 0 and 1. Yeah. Secondly, if you want to, I guess, see a demo on how to create, like, we want it to be like a 2x2 panel figure, I guess, where each panel is the gene body coverage plot for a single sample. This link might help you out here. And if this is a problem that people are having, I can do a demonstration probably Thursday. So yeah.
What's the difference between the plots for problem 2 and the plots for problem 3?
Yeah, there's no difference except that I think we really just want you to at least get Python running, because later in the course we're gonna have to do probably a lot more Python coding, and so we just want to make sure that, I guess, you know the basics of Python. So I think somebody's question I had a while back, I guess if you really don't want to make this plot in R, you can always say like, instead of giving it, you can just say in problem 2, for example, see my plot that I made in problem 3. But since it's so easy to make in R, it just does it automatically. You might as well just include both. But no, you're right, there's no difference really, other than that one's made in R and one's made in Python.
For problem 3, what type of data structure do you recommend we get the data into? For example, I try to use a dictionary and then I try to use an ordered dictionary, but it's difficult to then call index values.
That's a good question. I mean, what happens? So like, I know there's some sample names on it. What if you could you just read it in as a string and split that string up? Because
Yeah,
like, you know that each one's 100 long.
Yeah, so that's what I did. It's just that then I stuck it into a dictionary and trying to then call elements of that in the matplot structured format isn't working. So I didn't know if there was a better, so I've split the string, I've got those things. I just don't know what to put it into that's the best structure to use. Does that make sense?
I see.
I guess Panda can work better for this situation. So a package called pandas.
Okay, I'll look that up.
Yeah.
Yeah, so maybe especially Thursday, then maybe I'll actually get it out. I think the thing with Python is, firstly, I'm mostly an R user, but there's probably like many good ways to do something. But yeah, okay, so maybe I'll make a note here that Thursday, maybe I'll like do an example with like a fake data set. So yeah,
That'd be
okay,
great. Thank you.
yeah. Any other
Yeah,
questions about?
I also have a question. So for problem 3, yeah, I just mentioned I want to use like the package called pandas, but like the default version in R, it doesn't have the packages. So
I see.
yeah,
So
so
like
I need to.
So you can put, maybe it's, so you can specify the path to where your version of Python that has all these packages lives. So I've not tried to run Python in R Markdown that much. Like earlier, one option is just to do it on some other IDE that you use for Python. And then just to copy the script, put it into R Markdown and specify eval equals false. So like, if it's convenient for you to run it within R Markdown, do so, but it's not a requirement, if that makes sense. So like if you can't get like pandas to load, then just specify eval equals false in your chunk. Yeah.
Okay. And also for the gene body coverage in problem 2, can I just like do a screenshot and then put the figure in R Markdown?
Yeah, that should be fine.
Okay, thanks.
Sorry, when you say eval equals false, that just means the R Markdown won't run that code, right? What does that mean?
Yeah, so eval equals false means that when R Markdown, so like when it gets to that chunk of code, it will print the code, but it won't actually run it. So it's just like, that's kind of what we're looking for. We want to see your code. We want to see every like line of code that you did. But right, since it's like from a different language, and for example, like we're not going to have the files that you're like using, since you're not submitting that. It's fine just to like show us the code, but it does actually have to run within our Markdown.
Thanks.
I do want to just quickly show you guys how to use Salmon and RSAM. So these are both algorithms for RNA-seq quantification. And what that means is you want to measure, I guess, the level of gene expression that you have in your samples. And so for those of you who are in lecture today, so I guess my question is, what are 2 potential confounders of read count? If you're just looking at the raw, for example, in my sample, 50 reads aligned to this gene. So what are 2 potential problems with using that as a measure for gene expression? Or what's one?
Yeah, someone said in chat depth and gene length.
Yeah, so I think longer genes, you can see more counts from genes that are just physically longer in the genome. What's the other one?
Sequencing depth, I assume.
Yeah, library size. So like, for example, if you, in one like experiment, you see 100 reads or 100 reads mapped to this gene. And then the next experiment, 1,000 reads mapped to this gene. It could just be that in the second experiment, you had like 10 to 100 times as many fragments. And so for example, there's no actual difference. And so to get around this, like we introduced, I guess, these 4 measurements today: counts per million, RPKM, FPKM, and then TPM. And so counts per million are kind of the simplest, which is just like for a given gene, you're just looking at the number of reads like per 1 million fragments sequenced. Yeah, so for example, if you have 1,000 reads that map to the gene CD8, but you sequenced 1 billion fragments, then the counts per million for that gene would only be 1. Does that make sense? Yeah, and then RPKM, you essentially take the counts per million and then divide by the length of that gene. And then TPM kind of reverses the order, where first you divide by the length of the gene in kilobases, and then you normalize that, I guess, per million. Any questions about these 4 measurements that I can clarify here? And I think the lecture video that she included is great. So I'd suggest watching that again if you're confused. And so Salmon and RSAM are both algorithms that are going to get you these 4 measurements. Okay, so let's talk about Salmon. So Salmon is a pseudo mapper. So it's not actually going to like give you like a real alignment. It's really just primarily for measuring the amount of gene expression. So you're not actually going to get like a raw sequence alignment like you would with STAR. Okay, but let's briefly go over how you'd run this. Okay, so you can give it a FASTQ file as input, start a job, so go to a compute node, module load Salmon, and then the main command is salmon quant, so quantification. And then the inputs that you want to give it are, so the path to the index. So Salmon contains like a list of known, I think it's a list of known transcripts. It might be just exons. I'm not entirely sure, but the index is in the stat115 index folder. Next, you give it the path to the input reads, which are the FASTQ files. Number of threads. This will automatically detect if it's paired end or single end. I think these are single end for this problem. And then -O, so it's going to create an output directory. You just want to name that output directory, whatever you want. Okay, so I assume at this point, I think I've kind of explained like how to load the modules. So are we okay with this? It's kind of getting late in the PSET or the homework, so I don't know if anybody's done this yet, since it's still due in a couple of days, or like at the end of the week.
So sorry. So for this each line, do you just run it separately, like specify one line and then enter, and then specify the other one and then enter?
No, so it's like so it'd be like salmon quant space minus I, whatever, space minus R. So there's don't put any like new lines in. Sorry if that's a little confusing. But it's just like you would for STAR, where it's just like space. You just put spaces in between like the different options, no new lines.
Should the input be BAM files, or that seems to be what the homework is asking for?
So RSAM takes in a BAM file. I think we've included the BAM files and the FASTQ files, right? For each sample.
No.
We should have. I think so. In raw data 2, there's also a FASTQ folder, right?
Oh, maybe I didn't know that. I didn't see that.
Yeah, let me just check real. I think so. Like we've given you BAM files. CD FASTQ file. Yeah, they should be here. So we also have FASTQ files. Yeah, so the point of Salmon is that it's like very fast. You can just give it the FASTQ file, and then it so it doesn't actually like do alignment like STAR does. It just kind of approximately figures out where in the genome or where in like the transcriptome that your read came from, since it's just for for RNA-seq quantification. So yeah. So I think you want to give it a FASTQ file for this. And they're like they correspond. So like the BAM file that is like sample Y corresponds to the FASTQ file for sample Y. So yeah. I think you can give it a BAM file as input. Be interesting to see if that yields the same result. Probably will. But yeah, FASTQ as input. Okay, so what does Salmon output? So it's going to output a whole directory, but one of the files in this directory is called quant.sf, and it contains the expression levels, TPM, effective length, and then just read counts. So I actually have to compute FPKM because it's the version that we have isn't going to give you FPKM. Alternatively, you could try to install a newer version of Salmon where it looks like FPKM is like automatically given to you. But the formula is not too complicated. So I'd recommend just computing FPKM from the read counts and the effective length. Okay, and so let's just take a look at quant.sf. So it has like thousands of rows, since each row is like a transcript, and then the columns are going to be these measurements. So let's just look at the first 10 lines by typing head -10 quant.sf. So I did that, and so this is what showed up. So the rows here, these are transcript IDs. I think it's called ensemble transcript ID, followed by length in kilobases, effective length, TPM, and then the read count. And so these are all 0, so these genes aren't expressed. But somewhere in this somewhere in this file, there will be genes that are expressed. And part of the problem in the homework asks you to identify the gene with the highest level of expression. But to answer this question, I think we'd really prefer you not to give like the transcript ID in ensemble format. We really want to know the gene symbol. So for example, like CD8 is a gene symbol, and that we know, like I can kind of guess what that gene is associated with. If you just give me like an ensemble ID number, I have no idea like what this one is. And so you're gonna have to convert this to gene symbol. And one way to do that is with Biomart. And so if there's time at the end, which it looks like there will be, I can actually like show you how to use Biomart. But basically it's a database that allows you to convert between all these different like ID numbers. And so I think the first one here is 00004151118. And so I go to Biomart, which is here, and it's a database, and I put in that ID number. And then what it produces first is like, here are conversions to these other ID numbers and the gene stable ID. So then if I click this actually takes me to the gene for which the transcript came from. And so it's TRDD1. And then you can see like a description of it. It's associated with T cell receptors. You can see where it is. And there's even some more information. It's not very interesting in this case since this gene wasn't expressed at all. But when we ask like which gene had the highest expression, we'd prefer you to give gene symbol, not just ensemble ID. Yeah, so I can give a demo of how to like use Biomart. There's also a R package called Biomart. In case you, but since like, so this problem only asks you to do the one with the highest expression. So if you're just looking to convert one, I think Biomart, just like using the online database is enough. If you have like, if you wanted to convert everything to gene symbol, then using the R package is probably a better idea. Yeah, but I can give a demo of that at the end. Okay, the final thing is just how to run RSAM. And so the difference between RSAM and Salmon is that for RSAM, I guess, you first need to get a BAM file containing the alignments. But, so I'm wondering if you can just use the BAM file that's already given to you, now that I think about it. But I think the problem explicitly asks you to run star and then to run RSAM. So let's start with the FASTQ file, and then get the BAM file that we can pass to RSAM, which also does RNA-seq quantification. And so the command here is, and I got this out of the star reference manual that I attached earlier in the PowerPoint. But to give input to RSAM, so you want to call star, and then you have your default parameters here, which are like path to genome directory, FASTQ file. These are the parameters that we talked about earlier. But then you also want to add these two options, which are quant mode transcriptome SAM. So it's going to output a SAM file. And then the output, no, it's actually going to output a BAM file because that's what RSAM needs. And it's going to be sorted by location. And the purpose of this is just what type of file that RSAM is going to want as input. So you have to run this first, and then it's going to create a file called align to transcriptome out.bam, and you're going to pass that file as input into RSAM. Okay, and so once you do that, the main command for RSAM that you're going to use is rsam-calculate-expression. And so it can output a BAM file. We don't actually need that, so we can say no need to give us that. You want to make sure that you write --time, because for problem 5, it's going to ask you how long did this take. And if you do this, it's going to give you a file that like describes the total runtime and actually the runtime for like each part of the algorithm. So just make sure you do this for problem 5. BAM, so you're going to specify that the input is a BAM file. Number of threads. Input file. So this is the name of the input file that we obtained here in the align to transcriptome. There's also a reference for RSAM, which is in the stat115 index folder. And then finally, you can name the sample because it will also create an output directory. And below, I guess this shows like what a typical run would look like actually at the command line. So RSAM calculate expression, options such as number of threads, time. You're going to give it a BAM file. Then the input, which is the align to transcriptome.bam. The reference name, which is in the stat115 index folder. And then this sample name you can choose.
Hey, Philip, I have a question. So for the reference name, is that the path to file for the index? I did that, but then it actually didn't run.
Oh, so the index is an entire folder.
Right. I specified to that folder, but then I also look at the manual. It says it's the reference name.
So it's like the name of the kind of like the prefix. Because it says that the user must have run RSEM prepare reference before using the reference.
So are you specifying the absolute path to the reference?
That's why I did initially, but then I didn't run. So I'm just wondering why.
So like we've ran RSAM prepare reference for you, and we should have saved it in that folder. Did you run star first?
Yeah. I input the aligned.bam file.
Did you put them in the right order? Because order like matters here. So you need to
Yeah, I
input.
used the exact order like what you were showing.
I guess, did you load RSAM? Oh yeah, I assume you load it, otherwise it wouldn't give you an error message.
Yeah. It's okay, I'll try it.
Do you mind like, I guess, emailing me your bash code? Just somewhere where like nobody else can see it.
Sure, thank you.
But I'll at least take a look at the line, see if I can find anything. Okay, and so the RSAM output, it's pretty much, so it's contained in .isoforms.results, but it's basically the same as Salmon, except it actually gives you FPKM, so you don't have to compute it yourself, which is nice. Yeah, so we already talked about like what this file means with all the transcript IDs. It also gives you the gene ID, I guess, which is nice. Okay. Yeah, and I guess finally, I just want to quickly talk about like what an answer to problem 6 would look like, because it's pretty open-ended. I think it just says like compare like the various metrics that you got in problem 4. And so really, I just think, you just want to provide some plots, some sentences that like describe what you did. But for example, like one thing that you might do is ask if the metrics for gene expression are correlated, right? So like, I know there's like some people like TPM, some people like FPKM. You could ask like how across all genes or across all transcripts that we got from Salmon or from RSAM, are TPM and FPKM correlated? Also, we could ask, which one should I use, RSAM or Salmon? So you could compute the correlation between like the TPM from RSAM and the TPM from Salmon to see if these algorithms are really producing, I guess, the same results. Yeah, so I think like answering those two questions would be a good place to start, but also it's kind of open-ended. So I think if you have a question that you think would be interesting to pursue, go ahead and do that. One option is a heat map showing correlations between the metrics. Might be kind of a natural approach for this problem, since you can have like on each row is like TPM, FPKM, read counts, effective length. And then like each like grid, so it'd be like a 4x4 grid, and each square in that grid would have like the correlation between the two, the row and the column. That might be a good approach, but it's pretty open-ended. So I guess, yeah.